Welcome. I am Gio, your host for the Ask an ECV Expert series of talks. I am here today with Jeremy Rossman, virologist. Jeremy, please tell us a bit about your research and the work you've done in virology.
Thank you. So it's great to chat with you all today, and hopefully this is a good start to a long tradition that we can really learn from all of ourselves, because there's so much expertise here, and to be able to share that is fabulous. So I am a virologist, and for many years now, farther than I can remember back, I have researched viruses, cells, and the immune system, and how they all interact. So I did my PhD looking at immunology and how immune cells get turned on by viruses. I did postdoctoral research looking at influenza virus and trying to understand why the virus actually causes disease, how it affects cells, how it kills cells, and how we can circumvent that. And then I've run a research lab in the UK for about 10 years looking at RNA viruses. These are viruses that have a genome that's made of RNA, and we can talk about that later. But looking at how these viruses, such as the flu and Ebola virus, interact with cells, activate the immune system, cause disease, and what we can do about that, both in terms of understanding how that virus works on a really specific level, but also looking at how you can apply that to vaccine development, how you can optimize vaccine development, and then what happens in a real outbreak. When you have an Ebola virus epidemic in West Africa, that's a very different situation from what occurs in the lab. So I've also been conducting research on the ground, looking at how epidemic responses occur, what kind of integration we can get for behavioral science, sociology, anthropology, and biomedical science. And that led me to start a nonprofit that looks specifically at facilitating collaborations between scientists and on-the-ground humanitarian aid organizations. And that's what I've been doing ever since. So I'm happy to answer all sorts of questions on anything pertaining to virology, and I may not know all the answers, but maybe some of you will.
Awesome. Thank you, Jeremy. Let's see here. So I guess first question that I have for you is, I don't feel completely confident that I personally know what virology, the study of virology, really is. I get that it's research as viruses, but that doesn't really contain a good comprehension for me of, I spend my time doing X so that we can learn how Y works in reality. So how is virology different from epidemiology? What are other related fields? How do those different segments of the infectious disease science world all fit together?
So, I mean, this is a good question, because I just say virology, and that is literally just the study of viruses. So that could mean absolutely anything. And in fact, when we talk about what I've actually worked on and the kind of research that I do, a lot of it is viral immunology. This is how a virus interacts with the host immune system. Or sometimes it's host pathogen interactions of virus. And this means how the virus interacts with the host. Sometimes that's how it causes disease. Sometimes how it, balances that delicate interaction of what your body is doing and what the virus is doing. Sometimes it's what we call molecular virology. And this is the sort of really nitty-gritty details of saying, okay, we have this protein on the surface of the virus, and this protein interacts with this host protein, and it causes that effect. So it's this huge range, and really all of it overlaps. You can't look at host pathogen interactions without considering immunology. You can't look at immunology without considering host pathogen interactions. So they're all very related, but we do throw out a lot of terminology. So if I throw out some terminology and you don't know what it means, please grill me on it. I'll try to explain it. Epidemiology is the study of the determinants of health and disease. So this doesn't have to apply to virology. This could apply to any infectious disease. So the disease could be a bacterial disease, or it could be, something like diabetes. Or in fact, you can do epidemiology if you treat gun violence as a disease. So this is sort of looking at, you can do modeling, you can do, graphing of where disease are, factors that are correlated with it. So it's related. There are all sorts of nuanced fields that all come together to look at what we're looking at now, whether that's epidemiology or public health or molecular virology or molecular immunology.
Okay. So I think I understood that, and I'm going to relay back to you my interpretation of it. So epidemiology is the math of viral spread, roughly.
Or any disease, not only disease spread, but also the factors that lead to it. So, what are the risk factors? Where are they found? Yeah.
Right. And immunology would be something like how the body fights the virus.
Mm-hmm.
Host virus interactions would be something like what the virus does to the body.
Exactly, and what the body does to the virus.
Right. Which is a little bit more than how the body fights the virus, I would imagine.
Mm-hmm.
Because so I think I've got a good grasp. Thank you for kind of explaining the range of the field, because when you're not technically trained, all these things sound different, kind of mean something different. You can intuitively grasp some of it, but it can be difficult to really spot it down. And just reading stuff makes little sense when these are very dense, complicated technical fields with biochemistry, which is famously difficult to comprehend.
We do love our jargon.
Yeah, thank you. So next question is, we all know the basics differences, and that both bacteria and viruses can cause horrific pandemics. Bubonic plague, for example, is a bacterial disease, and smallpox is viral. What are the characteristic differences between bacteria and viruses? Are there any of these differences that we can leverage to fight either more effectively, either at the population level or medically? We know about antibiotics, but are there other technologies that are interesting there?
Absolutely. So, fundamental differences, first of all, bacteria can, not all, but a lot of bacteria can replicate by themselves. They can survive on their own. They have a lot of all of the sort of life-sustaining functions within themselves, whereas viruses are parasites. And I don't mean parasites like a worm or, something nasty like that. I mean parasites like they will not survive outside of your body. So, or somebody else's body or an animal or something like that. They're parasites. They need what you produce. And so that makes them fundamentally different. But one big difference because of that is that viruses tend to have very small genomes. Some of them only encode just a couple proteins. So bacteria and our cells have to have a huge number of proteins, have to do all of these functions. So they're very complicated. And because of that, it gives you a lot of targets, a lot of things to target with drugs or ways to disrupt it. But viruses are much more difficult because viruses only have a couple. And which proteins and how they work vary dramatically from virus to virus. So that's why we've had antibiotics for a long time, and a lot of these can target multiple different bacteria all at once. But the same thing hasn't been as true for viruses because it's hard to find those proteins and those functions of a virus that are the same from virus to virus and that we can actually target. But in fact, there are a few. And so some of you have heard of the drug remdesivir, which is one of the few drugs that we've heard about with some positive results recently, actually shortening duration of symptoms and not terribly effective, but moderately effective. This is a great example of what we can do by targeting viruses specifically. This is a drug that targets a protein that RNA viruses use. And so this is RNA viruses are viruses that have a genome that's made with RNA, whereas bacteria and all our cells, their genome is all DNA. But some viruses have RNA as a genome, and COVID-19, SARS-CoV-2, this is an RNA virus, just like Ebola, just like flu, just like polio, a lot of RNA viruses out there. And so we have some drugs that do target things that these RNA viruses have in common. Now, the viruses just differ so much from each other that it's difficult for them to work really well. So it's not the most effective drug in the world, but it does work, and it does work somewhat. So this is something that people have been trying a lot and has been somewhat successful.
So one of the things I'm hearing with the difficulty of treating viruses because of their small surface area is it's wise to not have viruses spreading, which is kind of the message of ECV. They're far easier to stop early in the spread than to treat after the fact, more so than bacteria. Is that sort of aligned with your understanding, Jeremy?
It's very hard to treat viruses without, I mean, we have a couple really good ways to treat them. We can, use a vaccine. Well, that's a great way to treat them. And, sometimes we can use antibodies and blood transfusions, and sometimes that's really, really good. But if you want to try and design a drug, like an antibiotic for a virus, yeah, that's very difficult and sometimes can work really well. But usually it's for one individual virus, and it takes decades to develop. So yeah, you're much better off trying to prevent it ahead of time.
Is it remdesivir that was originally treating AIDS that's been sort of a magical solution to that disease, or am I mixing up the drug here?
This has been used for a lot of different viruses over the years. This is not a highly effective HIV drug, but it's been used in a lot of different viruses. And the most recent one has been Ebola. And it does seem to help Ebola also. But none of these viruses does it seem to be a magic bullet. It's not like an antibiotic. And that's the thing, is these antiviral drugs typically are not antibiotics. They're not effective like that.
I have a question on the drugs side that I've been curious about personally. Is I know about quinine and the use as a prophylactic, and sometimes it's an early treatment, but it's not very effective as a late treatment, and it's much more effective the earlier you get it in your system, especially before you get infected. That has a stronger prophylactic and preventative, and then like early treatment effect that's more beneficial compared with like a late treatment, which is almost ineffective, completely ineffective with the diseases that that medicine treats. Is remdesivir much the same? Is it known to be prophylactic or just sort of early treatment?
Yeah, this is so true for so many different antiviral drugs. It's not just remdesivir. It's many. We have some very good antivirals against flu too. But some of these, so if you get flu and you might get prescribed Tamiflu. You tend to have like a 48-hour window from when you get infected to getting the prescription for it to be really effective. Remdesivir has a little bit longer activity. And in fact, one of the drugs that people are using now, dexamethasone, which is a steroid, that actually might be much more effective later because that targets the immune system instead of the virus. But yeah, for a lot of these antivirals, it's the earlier you get it, the more effective it is, but they're also not completely effective.
And there's definitely side effects to steroidal drugs that can be pretty terrifying, like weakening musculoskeletal tissue and other things. So steroids aren't a panacea, even if they're effective as a treatment. Uh, Kind of continuing with the questioning on the topic of the viruses. What are the differences between viruses? The DNA viruses, which you mentioned, RNA retroviruses, RNA viruses, which are different than retroviruses, coronaviruses specifically, how are they different? What are the lineages of these? Because I know most of biology, anything that's cellular has a single origin. That's a pretty strong assumption that we've made about biology, but viruses seem to have multiple origins. Would you go into kind of the history and lineage and differences between virus types? Because I think that would be interesting to unpack slightly here, not at length, but give us some. What are your thoughts on that, Jeremy?
Yeah. So, I mean, this is a huge and very complicated and very interesting question, but a virus is not a virus is not a virus. You have so many different flavors of viruses, and each one does something very different. And of course, the majority of viruses don't cause disease. That's also something that's important to remember. But you can, like I said, bacteria and your cells have DNA as their genome, but viruses can have DNA as their genome. They can have RNA as their genome. They can have just one strand of DNA. They can have one strand of RNA. They can have two strands of RNA. They can have one of DNA, one of RNA. There are probably other flavors of viruses that we don't even know about. So it just seems like if there's any variation that you can imagine, you have one of those in a virus. And if you haven't seen it yet, it's probably we just haven't found it yet, and not that it doesn't exist. So they're just incredibly complicated, incredibly diverse, and anybody wants to ask, diving in detail questions in there, we can go into it. But otherwise, I'm going to be talking all night just on, the differences of different types of viruses and how they work and where they come from. And it's fascinating, but it's huge.
All right. Thanks. So it's a long and complicated story to get into the variations of viruses. So what make coronaviruses unique? Is there anything that stands out there in terms of viruses, since this is a coronavirus that we're fighting here?
Well, yeah. So, I mean, one thing that makes it unique is the name. It's not completely different from any other virus, but if you look at it and you look at it by electron microscopy, and this is instead of using light to look at how something looks, you use a beam of electrons to actually look at it. And I'm not even going to begin to go into how that works, but suffices to say you can see things that are much smaller than you'd ever be able to see otherwise. And with that, the viruses look like the corona of the sun. So they look like this sphere, and they have this sort of fuzzy haze around it that looks like the corona of the sun. And some viruses have this look to various extents, but nothing quite like coronaviruses do. And this is just the structure of the virus. This is how those spike proteins on the surface look. And so that's a unique thing. But perhaps one of the most unique things about coronaviruses in general is that viruses tend to mutate very fast when they're replicating. This is because, when our cells are replicating, we have proofreading. So when we replicate our genome, we have proteins that go and look and say, did we make any errors? It's like photocopying something, but doing an error check and making sure no errors were introduced in that photocopying process. Viruses don't typically have that. And so one virus goes into one cell and makes hundreds or thousands of new copies. It doesn't have that error checking, and so you get mutations very fast. And this means you get evolution of the virus very fast. But for coronaviruses, it has a proof checking. So it is able to go and proofread and error check its replication. And this is really unique for viruses and really unique for RNA viruses. And thankfully for us, it means that coronaviruses mutate very slowly. And this is really good news if we're designing drugs, if we're designing vaccines, because it means the viruses tend to be more stable.
Yeah, that's good news for treatment, but one of the hopes that I've heard that's I think draws some upon the experience of the 1918 flu pandemic is that the disease will evolve towards reduced pathology because the selection pressures work that way, where viruses generally get less dangerous. But with this virus, it seems it would be as deadly for longer until we reach treatment capacity. Is that sort of a good assumption, understanding, outlook that I've presented?
Yeah, so it's all a matter from the virus's point of view, and obviously there's no, you're not implying intelligence here, but from a virus's point of view, all it's looking at is what allows it to survive and spread and replicate the best. And so if that involves causing more disease or less disease, then that is what tends to be selected over a long period of time. Now you have some viruses like Ebola. Ebola can kill between 60 to 90% of the people it infects. It's one of the most lethal viruses that we know of. It's not the most lethal virus that we know of, and that can be another question for later, but it's one of. And during the course of long protracted Ebola virus epidemics, so we had a Ebola virus epidemic that just finished in the DRC. We had a long one in West Africa in 2016. And even over a span of a year or more, the virus doesn't become less deadly. The virus is very good at maintaining itself and spreading itself because it forms such severe disease. So some viruses may adapt to get less virulent and less dangerous and cause less disease because it helps them to spread. Some might get more dangerous because it helps them to spread better. In the case of COVID-19, it doesn't really need to do a whole lot else. It's already extremely good at spreading. So we haven't seen a huge amount of mutations in COVID that have been selected for. We've seen one that has facilitated the virus transmit a little bit better, but we're not seeing a lot of this changing. The virus isn't getting more deadly or less deadly. It just seems to be very good at spreading as it is.
All right. I guess one of the questions that we've had asked here is, can you talk about the lungs and the immune system? I've heard the immune system is comprised of the innate and adaptive immune systems. Is the adaptive immune system present or less effective in the lungs? And I think more generally, I might throw in is the disease a respiratory disease or a blood vessel disease, which has been a point of discussion or gained knowledge in the recent weeks and months. So you can talk about that if you would.
Yeah. So, okay. So first of all, this is a respiratory disease. This is definitely a respiratory disease, but just because it's a respiratory disease doesn't mean it can't infect other cells in the body and doesn't mean it only causes respiratory disease. It's just, this is the primary site where the virus infects you and it primarily replicates. So this is because of the protein on the surface of the cells that the virus attaches to. So any virus needs to get into your cells. It can't do anything by itself. So it needs to get into your cells. And the way it does this is it basically has a key. And this is like the spike protein on the surface of the virus. But it needs to find the lock in order to open the door into the cell. And that lock is this ACE2 molecule that you've heard about. And this is on the surface of your own cells. And so whichever cells have this lock are the cells that the virus can get into and start infecting. And cells in the respiratory tract have a lot of ACE2. You have some cells that have ACE2 in the upper respiratory tract, in your nose, and the virus can infect and replicate there. Doesn't cause a lot of severe disease there, though. But your lungs have a lot of ACE2. Now, your lungs also have an innate immune system, and you have a whole body adaptive immune system. So every cell has some level of innate immunity. So it's got some way of protecting itself and trying to combat the virus if it sees a virus. Even your lung cells do. So this is part of that fight between the virus and the cells. But if the virus gets to your lung, well, then you get a very different effect than if the virus got into your nose. Because in your nose, there's a lot of cells there. It's a thick layer of cells in your nose. And if a couple of those cells die, it doesn't cause a huge effect. But your lungs are very thin. They have a very thin layer of cells because this is where oxygen has to absorb across. So you can't have this thick layer of cells. So if you have a very thin layer of cells and then those cells get infected and start dying, you can damage your lung very quickly. Also, if you have all of the immune response causing inflammation, maybe fluid buildup, if that happens in your nose, well, then you have a stuffy nose. But if that happens in your lung, you have pneumonia. And so it's not a completely different mechanism of what's happening in the upper respiratory tract or the lower respiratory tract. It's just that your body has a lot less tolerance for damage to the lower respiratory tract than it does to the upper respiratory tract. Now, it's not just affecting the respiratory tract, though, because you hear about blood vessel disease, you hear about neurological disease. So again, that lining between the air side of your lungs and the blood side of your lungs is very, very thin. This virus is getting in there, it's killing those cells. And so that allows the virus easy access into your bloodstream. So the virus can get into the bloodstream, and your blood vessel cells also express ACE2. And so that allows the virus to infect your blood vessel cells and to spread from there. But also your immune response is being turned on across your whole body because it's a big infection. And the immune response causes issues throughout your whole body. I mean, fever is an immune reaction, but you're not just getting a fever in your lungs, you have a fever in your whole body. And so this is additional reason why, even though it's a respiratory infection, you can start getting symptoms throughout your whole body.
And that's pretty terrifying.
Yeah.
What you were saying about innate and adaptive immune system brings up one of the core points that we at ECV has been pushing about masks, which is any mask is kind of better than no mask. There's definitely more effective masks. But with innate and adaptive immune system, the innate immune system is very sensitive to viral load, which we know from healthy medical personnel who have a high viral load often get sicker than even unhealthy people who are exposed to less like chronic viral load. So would you go into viral load and our understanding of that for this disease?
Yeah, so there's viral load, and then there's the amount of virus that you've been exposed to. And viral load is how much virus you have in your body at any one time. So if somebody does a diagnostic test to you, then they can often tell you how much virus you have. And the more virus you have, it does tend to roughly correlate with how severe the disease is, but not always. It's not perfect. But then you have the amount of virus that you're exposed to. So if you're outside and somebody coughed a minute ago and then you walked through it, the amount of virus you're going to be exposed to is very small. But if I'm sitting right next to you and I don't have a mask on and I sneeze in your face, that's nasty, but it's also a very high exposure. The amount of virus that you could potentially breathe in is very high. And the issue is you're just, if you look at the curves of the number of positive cases in the US. It's an exponential curve. So if you think of your body as the US, well, if you have a very high viral load, you're starting on that exponential curve further up on the curve. So you get much sicker, much faster, and sometimes that allows the virus to overwhelm the immune system before you can start to combat it. Whereas if you start very low on the curve, the same thing is true in your body or a case, flying into the US. If you only have a couple cases and you're doing really good surveillance, chances are you might be able to stop it before it really spreads and before it gets severe. Same thing is true. If you start with a lot of cases all at once, it's going to be much harder to control.
Right. And another point on masks that I think may be important to discuss is droplet versus aerosol and how the disease physically manifests itself. Because my understanding is any type of mask, even something as hilariously misbegotten as mosquito netting, would be effective at preventing spread, not protecting you so much, but preventing spread, because the airflow of the droplets will be disrupted by something like mosquito netting, causing them to drop more, meaning they're less in the air. And this is a droplet spread virus, which means that it's the larger moisture particles where the virus resides in. And that means that it's really, really easy to prevent spread by disrupting the exhalation of the virus particles. And that's my understanding, but I'd like to get your perspective on this and some of your deeper knowledge on it.
Yeah, so, we hear about a lot of terms. We hear about, aerosol transmission and droplet transmission and airborne viruses. And all of these are actually very, very similar things because both aerosol and droplet are a form of airborne transmission. The only difference, and aerosol transmission is also droplets. They're just much smaller droplets. So all of these droplets, and what we mean by droplets is just a little bit of fluid that comes out of your nose or your mouth that has a virus inside. That's all a droplet is. It's just very, very simple. But they come out of your mouth or your nose in all sorts of sizes. So some of them are really big, some are really, really small. The smallest ones are what we call aerosols. And these really small ones are very difficult to prevent the spread of because they can pass through masks very easily, not medical grade masks, but a lot of masks they can pass through easily. They can come out of the sides of masks and spread over large distances. They don't tend to sediment down to the ground as easy. They can stay in the air for a long time. These are the particles that can spread through air conditioning and ventilation systems and things like that. Thankfully, it looks like COVID-19 is mainly respiratory droplets. There's some evidence that maybe it forms some aerosols, but it tends to be more of these heavier, larger respiratory droplets, which is why any sort of face covering is effective. Now, the thicker the fabric it is, the better. If something has a lot of spaces or pores or just one, they did a study recently that was looking at just one thin layer of a bandana, and a lot of those respiratory droplets went through it. So the thicker, the better, but this is respiratory droplets, and face coverings, even non-medical grade, are effective because they just really disrupt that. But we don't, thankfully, get those long distance, tiny aerosol particles that just float around for a really long time that can spread through ventilation systems, thankfully.
Yeah, and so it's a combination of having some innate defense that means that there's a certain threshold for the viral load, correct? So it may not be one virus that infects you, but 10 would, or something with, there's a certain threshold, and that might vary person to person and such. But it's not a all or nothing. There's a sort of gradient of effectiveness, and having something between you and the virus is a good idea, correct?
Exactly. It's not all or nothing. It is definitely a gradient effect, and the thicker and the more robust the barrier is between you, the better and more effective it is. And yeah, in terms of the number of viruses, we don't know how many individual viruses it takes to cause infection. This is definitely going to vary from person to person. We also don't know how many viruses are in most respiratory droplets. This is something that we don't know for a lot of viruses that have been around for a really long time. And we only have a couple months. It's shocking that we know what we do about COVID with such a short span of time. So yeah, we don't know. It could be just a few viruses. It could be, hundreds of viruses. And again, it's, gonna vary from person to person, I'm sure also. And it could be, a certain number of the really large, heavy respiratory droplets, or maybe even fewer of the smaller droplets. And so we just don't know.
Right. Speaking of what we've learned, it's been 6 months since COVID arrived and started spreading in the US. What have we learned about this disease in that time that really stands out? What is sorting itself to the top of the knowledge stack? What should we all be aware of about this virus?
That is everything. Given the fact that we didn't even know this virus existed a couple months ago, what we're learning is, a daily challenge and, a daily triumph. And it's everything. We're starting to learn that the majority of people do develop some level of immune response, but that people that have mild or asymptomatic cases, might not develop a robust enough immune response to really protect them. We don't know for certain, but it seems like that might be true. We know that the immune response tends to last a little while, but it also seems like it fades within a couple months after being infected. So we don't know how long it's going to last. We know the sort of comorbidities, the other diseases that you have that make people really susceptible to severe COVID disease. We now know so much more about that, hypertension and obesity and diabetes, and the effect that these diseases have in making COVID disease so much worse. We know that there's a tremendous number of people that never show symptoms or show very, very mild symptoms, but these people can still transmit the disease. Children is a huge area that we're starting to learn, because we're learning that in a very small percentage of cases, children can have life-threatening disease, but the majority of children don't show any symptoms whatsoever. And in fact, the majority children seem to be infected at a lower rate than adults. So they don't seem to get the disease as much. And if they do, they don't seem to have as much severe disease. So we're also learning the huge breadth of symptoms that you can have. We're learning about the cardiovascular disease. We're learning about the blood vessel inflammation. We're learning about neurological disease. And we're learning that for many people, just clearing the infection doesn't mean that you've resolved the symptoms. That in a lot of cases, people have symptoms for months after clearing the infection, that it's not a simple, you're done and you feel better. So
that's terrifying to hear. That again, even after you have no virus in your body, you still have chronic symptoms that last far longer than the infection. One of the things that I've believed that I've learned about disease in general, and this disease in specific, is that when we talk about immunity, what we are actually talking about is not a binary of immune, not immune, but of a resistance threshold. In other words, you gain resistance to the disease, and we call that resistance immunity if that resistance is higher than what your natural exposure to the disease would be under most circumstances. Is that roughly an accurate depiction of the terminology?
Yeah, it's a gradient. And again, this gradient is, you know, it's not just you're not immune or you are immune. Everybody has different levels of how immune they are, but then this is a complex interplay because, okay, do you have any other coexisting disabilities that might predispose you? Or how much virus were you exposed to? If you got a huge viral exposure, then you might need more of an immunity and a pre-existing immune response to be able to combat that. So it's all a gradient and it's all individual levels. And we don't know how much immunity, for immunity takes a lot of different forms, but one of the forms is antibodies. So we don't know what antibody levels you need in order to be protected against COVID infection. So we don't know that in general. And then we don't know how much that varies if you have a big dose, if you have a small dose. So there are so many parameters here, and there's so much we don't know yet.
Right, right. One of the questions that is philosophically interesting to me, but I think we've somewhat covered, but I'd like to get a more philosophical answer to is one of our guys here has asked, so you can develop antibodies immunity to a virus with exposure without infection, like a brief exposure or a very, very low grade long-term exposure. So you never actually have the virus replicating in your cells, but you build an immune reaction to that. Is that possible with viruses in general? I think we've kind of clarified that COVID itself, those low asymptomatic exposures seem to be not nearly as protective as a symptomatic exposure that's been recovered from. But philosophically, is that a valid perspective on a disease?
So you never get immune response building up if you just encountered a virus. It always takes an infection. But all of these asymptomatic cases, all of these mild cases, all of this that we're talking about, those are all infections. The virus is infecting your cells, it's replicating in your cells, it's probably even killing a lot of your cells, but it's either held in check by your immune system or it hasn't been replicating enough to cause enough disease that you notice it. You know, on a cell level, the virus is causing disease and it's replicating. That's what it's doing. But even if it's not causing disease that you know of, you can still be building an immune response. And this absolutely happens. Although the more severe the disease is, the more your immune system gets activated and you get a fever and you feel really tired and you're not feeling very well. This is all your immune system being activated. And so the more your immune system is activated, the stronger the immune system, the immune response is being developed. So you can develop one through very mild infections, but often it's not, it's a milder immune response and it's not necessarily protective. Now, we don't know for COVID. We don't know if that mild asymptomatic infection is in fact enough to generate a protective response. It might be, but it doesn't look very promising on that side for now. The best thing that you could do is have a vaccine that gives you that strong immune response without a lot of symptoms, and then you get the protection.
Yeah, because one of the things that I had seen is as a potential, like in the herd immunity space, is as a potential idea is something that you might call COVID jousting or COVID chicken, where you want to expose yourself just enough to get infected, or not even to get infected, just enough so that you get the immune reaction without getting infected. But it seems that that is a foolish errand, given that any infection can be fatal. And while viral load is definitely the best strategy is definitely not to get infected, because there's no Goldilocks zone, so to speak, which is sort of the underlying philosophical idea there. I mean, not to go too far into it, I think, but if you want to say just a bit more on herd immunity and sort of the related things, this would be a good time.
Yeah, I mean, this is an interesting thing, because we talked about that, how much virus you're exposed to and what kind of disease it causes. And so if you come out really early on that exponential curve, then your body has more of a chance to combat it before you get to the exponential phase and really start to get severe disease. So you do have techniques, and this is the original form of vaccination, variolation, which is, what they used to do for smallpox, which is, you could take some infected scabs or sores of that infectious material and give just a little bit to a person, and give it to a person in a part of the body where they wouldn't normally be exposed to. So, this is why, you give it to them in the muscle of the arm. Now, if you did that for COVID-19, well, you have some risk that maybe it gets into the bloodstream and stuff like that, but it's going to take a while to get from your arm to your lungs. So, that gives you a little bit extra precaution, and you're starting with a very low amount of virus. So for diseases when you didn't really have anything else that you could do, it was a way to help. But the problem is, a lot of people that would get that ended up getting full-blown smallpox and dying. And the same thing is true is for COVID-19. Yeah, I've actually heard of people talking about, can you give somebody just a very low dose of COVID-19 in a controlled environment, and then hopefully they build enough immune response and are protective. But we don't know why some people get really severe diseases and other people don't. Yes, if you have hypertension and diabetes and obesity and are over 80, you're at a very high risk. But a lot of healthy, young or middle-aged people get critical illness, and we don't know why. And so this is always a risk. We don't know what threshold you need to get infected. We don't know what threshold you need to build protective immunity. And we don't know who's gonna have really severe disease and who's not. So the risks are just really, really large for that. Now, if we had a strain of COVID-19 that we isolated from people that was maybe related but didn't cause disease at all. I mean, if you could immunize somebody with one of the seasonal...
The cowpox of COVID-19.
The cowpox of COVID-19. And maybe that's one of the seasonal strains of coronavirus. It doesn't look like it, but, if you could do that, then you might be able to, then that's the real first vaccine. That is how you could vaccinate somebody, and that's an attenuated virus. This is what we have for some flu vaccines and a lot of other vaccines, and it can be very effective.
Okay, we've got a question here, which I think might be to elaborate on cytokine storms, but it seems COVID-19 damage is mainly due to its triggering of the immune response. Can we go into how the immune response affects a mild versus severe infection? And is there some knowledge about upper and lower respiratory tract infection and how this might have implications for treatment?
Yeah, so cytokine storm is one of those other terms that we've all been hearing a lot about. This is a very general term, but what it basically means is a hyper response of your immune system. So your immune system normally gets triggered by, say, a virus, and it turns on, and you get inflammation, and a lot of cells are activated. You get things like fever, and you're tired, and maybe your muscles ache a little. You get a lot of different symptoms that are all from this immune response. But sometimes that immune response responds extra strong or doesn't shut off appropriately. And in those cases, you can get this feedback where you get a really strong immune response, and sometimes that immune response is enough to cause severe or critical disease itself. So this is what we're talking about, cytokine storm, because COVID-19, it can damage the cells that line the respiratory tract. And if it does that in the lungs, it can cause some disease. but it looks like the majority of severe disease that we're seeing is coming from this hyperimmune response. And this is what we see in children that have the pediatric multisystem hyperinflammation syndrome, because they're also getting this really strong immune response that's not shutting off appropriately and is causing damage itself. So that's, this is also why that steroid, dexamethasone, has been shown to actually increase survival rates for the very severe cases. Now, you wouldn't want to give that to everybody. You wouldn't want to give that to people that maybe have just an upper respiratory tract infection where it's not causing so much severe disease. You don't have pneumonia. You're not at a greater risk because the immune response normally is helping your body. But if it doesn't shut off, if it's too strong, if it occurs in a really delicate tissue like your lungs, then it can cause a lot of damage. And this is why shutting it off with a steroid like dexamethasone can actually improve survival. So it's this balance. You want the immune response because that's the only way you're going to clear the infection, but too much of it can also cause damage.
All right. So I have a question. Any provisional understanding of what qualities might lead someone to being asymptomatic? And what is asymptomatic really when there's pre-symptomatic and subsymptomatic? And is this a useful term? Because this is definitely a point of broad confusion. So what is your view on asymptomatic cases?
Yeah, I mean, it's a hugely good point in terms of the terminology here, because we're hearing a lot of things and often they're all grouped together or they're used interchangeably when they shouldn't. Asymptomatic means that for the duration of your infection, you do not have any symptoms. So you clear the infection and you've never had any symptoms. Whereas if you're looking at somebody at one specific point in time and saying they have the virus, but they don't have symptoms, well, you don't know if that's an asymptomatic case or a pre-symptomatic case. And where you define symptoms also depends, this is in a lot of cases subjective. How much is somebody paying attention to changes in smell or taste? Well, do they know to look at those, to report those? Because we can't measure somebody's changes or smell or taste. We can measure fever. There are a lot of things that we can, but it's this complicated thing of, how accurate are we detecting all the symptoms? And then look at this over time. It's not just enough to look at one point in time. So that's the difference between pre-symptomatic, no symptoms right then, but you get some before you cleared the disease, versus asymptomatic, you don't have any symptoms at all. And those are really the only two sort of relevant terms in this. In terms of why, we have absolutely no idea. Could be how much virus you're exposed to, could be your immune system, could be something else. Absolutely no idea.
So I guess asymptomatic has no meaning in the world of PCR testing because there's no differentiation.
No.
So you would have to negative on a PCR test and positive on an antibody test to have a definition of asymptomatic that works in the current testing space. Correct?
And ideally, you would get PCR tested, show positive, not have any symptoms, then go a span of time and then have an antibody test and show antibodies being developed. And then at no point in time did you have any symptoms. That would be the best definition of an asymptomatic case. But it's really hard to get that rigorous data on asymptomatic cases. And the data that we have, it looks like it's not a lot of cases that are actually genuine asymptomatic cases. A couple percentage, whereas a large number are either pre-symptomatic or just extraordinarily mild.
Subsymptomatic, yeah. So that's kind of useful clarification, I think, is we can't define asymptomatic given our testing technology without having several hurdles, because you'd sort of have to have a positive PCR, a negative PCR, and a antibody, or at least a PCR, no symptoms antibody, because once you have antibodies, your body is at least fighting the disease if it hasn't overcome it and cleared yet. But I think you need a negative PCR to clear, which I think concludes that. So it's kind of a non-term in the, like, it's not an accurate term. It might be a precise term, but it's not an accurate term given our current testing methods. And then also it adds a lot of confusion, given that people assume that asymptomatic means you can walk around if you're not spreading the virus, and there's an association of the spreading the virus with not having symptoms or symptoms. And so it just compounds confusion. Well, it's not even, I don't know if it's not technically, but it's not functionally accurate, given the way that the testing works.
Yeah, I mean, you don't, if you had the positive PCR followed by the negative PCR and no symptoms, even if you didn't have the antibody test, you could still make that assumption. But yeah, everything that we're talking about right now is based on, the single test. And then do you have symptoms or do you not? But you can't tell asymptomatic versus pre-symptomatic at that one static point in time. And so yeah, it's not really a relevant distinction. And it doesn't matter in terms of public health, because what matters is saying, if somebody doesn't have symptoms, can they still transmit the virus? And the answer is yes. It doesn't look like they transmit it as well as people that have symptoms, but they can absolutely transmit it. And that's the crucial point.
All right. Question we have, that's one that I'm very interested in, is I've heard of different blood types, say type O, have milder infection or don't get the infection. Why does blood type differ in this? Why does blood type matter, or does blood type matter? What do we know about blood type and COVID-19?
Yeah, there does seem to be a correlation between blood type and severity of infection. Why that is anybody's guess. It looks like there may be some correlation with the proteins that the virus uses to cause the infection, and that the blood types that have the stronger correlation have more of it. But this, right now, we're at the point of saying, okay, in a very small collection of data, we've seen that does appear to be a correlation. We don't know if this is going to stand up to a larger data analysis. And realistically, we have absolutely no idea why. And if, one predisposes to more of the sort of blood vessel type disease versus a respiratory disease or anything like that, we just really have no idea. This is one of those, well, this is interesting, and we'll probably have better data in a couple weeks, a month, tomorrow. But yeah, this is one of those odd things that we just really don't know yet.
All right. There's a group of questions, one of which we have in the chat, which I definitely wanted to get into, so we'll go with this, which is, what are the chances of symptoms showing up years later? What are the long-term implications of this disease, given what we know about how it works and comparing that with previous diseases? What could be the long-term effects that we just cannot predict and cannot rule out at this point in time? Are there things that we have been able to rule out in terms of long-term dangers of this disease, and all of the related things about the hidden tail risks that the people in our group, who are all fans of Nassim Taleb, or some at least are fans of Nassim Taleb, know about, that most people have never heard about or thought about before?
Yeah, That's a great question, and I would love to be able to answer that. The short answer is nothing, anything, and anywhere in between. There's no really good way to know that. Every virus tends to be unique, and every person responds to a virus uniquely. And the issue is that we have so many people infected with this virus that even if a long-term effect is very rare, chances are we're going to see it. And this is the same thing with the pediatric syndrome, is the numbers are really small. This is pretty rare. And if it was a small outbreak or an outbreak within one country, we might not see any cases of that. But with this many infected, we start to see even really rare symptoms. So chances are we will see some long-term effect of the virus coming back up later. What that is not clear. This could be neurological symptoms. We've seen a lot of people have neurological symptoms that can be things like Guillain-Barré syndrome, things that involve neurological damage. We've seen encephalitis. Some of this seems to be immune mediated. Some may be caused by the virus. With some of that, there's always the potential for this to persist and then to be sort of reignited later. So one example is there's been antibodies that people have seen against being developed during the course of infection against what we call phospholipids, part of the membrane on your cells. Now, this is not normal. Normally, you don't form antibodies that react to yourself because that's autoimmunity, and that can cause a lot of disease. So they're seeing this in some people, and we don't really know what kind of disease it's causing, but it's possible that that could cause disease over the long run. It's also possible that some of the immunity that your body makes to this disease could be activated by something else at a later date. Maybe there's another coronavirus that has some sort of cross-reactivity to this virus. And so you get some immune response, later on you get infected by a different virus, and that causes a really strong immune response that causes damage. I'm purely speculating here. There could be anything, but from coronaviruses in general, this is not a family of viruses that tends to cause long-term reactivations or long-term disease. And what it looks like for people that are sort of long-term sufferers of coronavirus is not like a reactivation that happens later, but that the damage caused by the infection is taking a long time to heal, and sometimes the immune system is taking a long time to shut off. But that's different from saying you recovered, you're fine, and then you have a severe disease later. It's possible, but we don't really see that much with coronaviruses. We didn't see that happening with SARS. So it's possible, but I think it's unlikely. But I think the more likely thing is just that the damage caused by the severe disease will take a long time to fully heal.
So a friend of mine, Brett Weinstein, talks a lot about the sort of, you have a lifetime budget of cellular healing. Kind of your telomeres have a certain number. That's the number of cell divisions that your cells can go through. And so damage kind of takes out of that lifetime budget. And so one of the points that he makes is an infection is not just, I was coughing for a week and now I'm better. It's I've had to pay, and maybe a very small amount, but I've had to pay to repair the damage done, which will be taken out of my lifetime. Now there may be some counter effects where you need a certain amount of exposure to disease to be healthy long term, which is sort of the dirt hypothesis of health. Sort of, we've not had enough exposure to pathogens in the modern world, so our immune system is dysmodulated. And I'd be curious about your thoughts as a virologist on that complex of thinking, and if there's any part of that that might relate to the COVID-19 situation beyond the getting sick is probably a bad thing, so take it seriously point, which is core to what we're trying to say in general.
Yeah, I mean, we had, in terms of what's actually happening in your body, how your lung tissue is regenerating, how long your cells will survive, if the extra lung repair that you need to do tasks your stem cells so that they can't repair along it, we don't know. You can look at one individual cell in the lab and tell that, and you can get that information. But in people, we just have no idea. So we don't know if an infection is actually, if you get a lung infection, it's obviously bad and it takes a long time to repair and recover from, but we don't know if that's going to permanently sort of decrease the longevity of the lung cells that you have there already. It might, we just have no idea about that and have no real way to tell that at all. But we do know that you need your cells to repair, and that takes a long time and also has risks. Your cells are replicating, they're repairing, and if there's an error there, you can get problems. But overall, this is what your body does. Because even if it's not something huge and severe like COVID-19, your body is constantly repairing your cells, and your cells are constantly being damaged. And this is what your body does. And for the most part, it does it really well. So I would say, don't worry about that. First of all, there's nothing you can do about it. Second of all, we just don't really have that good understanding of sort of long-term regenerative capacity of tissues. It would be great to understand that more, but we just don't know that yet. And on the other point, the sort of, this idea that, we're the hygiene hypothesis, the idea that we're living in a more sterile environment, so we're not exposed to as many pathogens, and so our immune system changes. This is absolutely true. There's no question about it. And again, it's not, we're not saying, okay, you should go out and, go get infected, because often we're talking about, say, asthma compared to perhaps a life-threatening illness. So it's not a direct comparison, but our immune systems are designed to respond. And it's designed to do that all the time. And if it's never used, and that's not true, it's always used, but if it's not getting that sort of regular use, and specifically for parasites, and that's the thing, is it's not really that we're not getting exposed to bacteria or viruses. It's just that in, say, the US today, most of us don't have a lot of parasite infections. And the way the body responds to parasites is actually very similar to what happens in allergy. It's that sort of same mechanism, which is different, a different type of immune response from what happens with viruses. And so if you're not getting that sort of regular use, then small little triggers like pollen can sometimes trigger a really strong response. Whereas if you had, a lifetime of parasite infections, your body doesn't respond in the same way. And this is something we're sort of really just figuring out right now. It's unbelievably complex because there are so many interactions between the different arms of your immune system, the different parts of your immune system. What happens in your immune system in childhood affects how your immune system responds as an adult. So it's unbelievably complex, and we're just starting to tease out some of that.
Yeah, it's the immune system is at least somewhat anti-fragile, and so this is something I've been curious about from someone who's technically trained, because a lot of this stuff I've heard from people who are a little bit more anecdotal and kind of in the ancestral health field, which I think is the baby steps of evolutionary medicine, which I think is a interesting field that we haven't really developed as humans yet, but is definitely nearing the horizon of our knowledge base. But that was, kind of a personally interesting question. But speaking of pediatrics and children and learning and human knowledge base, what are your recommendations for reopening schools, particularly elementary schools? We are in the middle of summer, so this isn't immediately urgent, but it looks like COVID is going to be around a lot longer than we had hoped at the beginning. And so what should we be doing to prepare for the regular school season, and what can we do to stay safe with long-term COVID in society unless we have a major policy turnaround in the near future?
Yeah. So, I mean, this is, such a prime area that everybody wants to know and nobody really has a good answer for yet, both on the policy side as well as on the virology side. So the issue is, first of all, we're viewing schools from a sort of historic point. So much of what we're looking at for COVID-19 is stemming from sort of our best analogy, which is the flu. Because the flu is what's caused the most previous pandemics, caused the most recent pandemic, the 2009 H1N1 swine flu pandemic. So this is what so much is based upon. A lot of the discussion of herd immunity, a lot of the discussion of fatality rates, a lot of the discussion of schools. Because with flu, schools were one of the most robust ways to transmit virus into the community because children tend to spread the flu very, very well. Now, with COVID-19, it doesn't seem like that's the case. Children, as we talked about, they don't seem to get infected as well as adults. They don't seem to spread the virus as well as adults. So, and they don't tend to have as strong of a disease as adults. So that's all on the plus side for reopening. But the risk is we just don't understand what real role children have in transmitting. There have been some studies that said that children are typically not the ones that bring the virus into the family, even if they're going to school. And so, there is a lot of governments are making that argument, saying that children don't get infected as well. They don't seem to spread it to their families. And so we should, reopen the schools. And there's a strong argument for that. But the issue is we just don't know quite enough yet. We don't know if children aren't bringing the disease back to the family because they've been isolated with their family. The adults might be going out to the store, but the children are staying home more. So the exposure of the child outside of school, if schools are closed, the exposure of the child is much different. So that could be playing a role. Also, you're having adults, you have support staff in the schools. What role are they going to have? And even if children aren't spreading it well, you put enough children together in a small environment, and they are going to spread it. And then in a small number of cases, children have severe disease. You don't want to be creating a situation where we're starting to get spikes in this pediatric hyperinflammatory syndrome. Even if children aren't spreading it well, a little bit of transmission can start to bring it back to the community. And then you could say, well, maybe you could have physical distancing in schools. The kids could wear masks and things like that. And that is absolutely true. You can set up schools differently to be more protective, and that can be effective. But you can only do that with certain age groups. Trying to get, a bunch of kindergartners to wear masks appropriately and so physical distance from each other, good luck there. Maybe by 5th grade, you might be able to do that. So we just don't quite understand that how the disease is spreading amongst children. And the other thing that's really important is what is the community level? We can't just talk about this as a group, reopening schools for the USA or reopening schools for any one country, because what's happening in any one city where the school is going to be dramatically different. So if you have extraordinarily low numbers of cases and you have a school that, is able to add some physical distancing, maybe do, only half classes, get the kids to wear masks, reduce the support staff, things like that.
Perhaps a boarding school, like, those are more popular in the UK, is my understanding, but that might allow you to have isolation that would make it a safer operation. There are definitely things that can be done, correct?
Yeah, I mean, that's the thing. There are things that can be done. You just have to do it carefully, and you have to make sure that it's in an area where the disease is quite in check. Because if we get to a level in, any one city in the US where the disease is sort of like we've seen in South Korea or a lot of countries that have gotten it under control, where it's, just a very few number of cases, then if you add these precautions to schools, you might be able to actually reopen it safely. But if you try and reopen schools even with the precautions in, Texas right now, you're just asking for trouble. So it's got to be context dependent. It's got to be approached with caution, and ideally, we need more data. So
Yeah.
this is a very dissatisfying answer to give, because overall the risks from school are lower, but children and schools can be really effective ways of transmitting viruses to multiple families in the community that can then spread from there. So we have to approach it with real caution, but it is a possibility if we do it really carefully and in an area that has good control already.
Massive testing, quarantines, the stuff that we talk about as an organization in general is necessary in order to have the general security in the community to allow for schools to be open safely, where you don't cause a shutdown of the entire local economy a month later. And that I think should be the real concern, is not just, it's the school that causes, then your kids get sick. It's what happens if you reopen the schools and that causes a new outbreak in the community level, then all of a sudden you have to shut down everything again. So it's a big risk for everything, not just the schools. So schools can't be separated from the community like that, correct?
Exactly, and it's all a gradient. It's not that children don't transmit the virus. It's not that children, can't spread it through the community. They do. The children absolutely can spread it between themselves and can spread it to adults. They can. It's a risk, but again, it's a much smaller risk than adults. And so this is all about risk management. And, we're saying, okay, if you wear a mask, well, that's better than not wearing a mask. But if you wear a mask and physically distance, that's even better than just wearing a mask. So this is all a gradient. There's no, critical threshold where all of a sudden it's safe. If the coronavirus is still around, it's never going to be perfectly safe to open the schools. But it's not perfectly safe to open businesses right now. So it's a matter of saying, do we have enough data of how well the virus transmits in children, which we're getting, but we're not there yet? And then can we make sure we take the precautions of saying, what is the level of virus in the community? Do we have the requisite infrastructure of testing, contact tracing, isolation? Do we also have the right infrastructure in the schools and the proper protections in the schools? And if you can add all of that together, then you can start to design a safer system. But there's not any one thing. There's not any one answer.
Right. Okay, so I have another class of questions which I think will be really interesting to go in. So when I first started to discover COVID and started doing my research and started contributing to the endcoronavirus.org effort, R0 was a big thing. This was February and March. And it took me a long time to realize that some of the use of terminology was kind of confused. There's R0, which is what I would call a raw spread rate. So that's the spread rate without additional constraints. And then there's something like R effective, which is what you currently have in the population. And so what you can do is you can change the constant. It's not really a constant, but it's closer to a constant of R0 with different social and medical measures to reduce the R, which is the reproduction rate, to something that is effectively below 1 for the community level, for the population. And that will mean that the virus will eventually go extinct in the community spread because the virus is reproducing, it's spreading slower than it is being overcome by the immune response of people. And then also, although it's kind of tragic to say it, people dying and no longer being infectious because of that. And so what are your thoughts on R0 and R effective and kind of spread rate and population stuff? And then also there's a question that we'd have, which is, do we have a profile of a typical super spreader? Or are super spreaders more of an event in a particular point in time, which is unique? And so what are super spreaders in relation to R effective? Because we kind of have learned that most spread events are like one person spreads it to half a person, and then one person spreads it to 1,000 people. And you get either or situations far more than it's like, most people spread it to 4 people if the RO is like 4 people or whatever. It's much more, there's a very, very fat tail, to go back to our risk aware language.
Yeah, so I mean, this is a really interesting question, and getting into the super spreader is really, I think, really important. So just on the basic sides, R0, this is the sort of basic reproduction number of a virus. This is basically, if I'm infected, on average, how many people am I going to spread the virus to? But this is an average. And this is an average at sort of the start of the infection, with everybody being susceptible to it and no precautions being taken. This is sort of, on the virus side, this is the best situation for the virus. So we have that. But that is, just one sort of static number. It doesn't reflect the current situation. The current situation is this R effective, which is RE, or RT, which is R over time. And this is basically what the current situation is. And this is how many people I'm going to spread the virus to, given how many people in the population are now immune to it, given the public health precautions that are being taken, and all of that. And then the idea is that if that R effective is less than 1, well, then that means the virus is going to stop spreading eventually through the community, because I'm not giving it to every one person. So it's not propagating at all. It's starting to die out. So that's, from a public health standpoint, that's what we've wanted. We've wanted this R effective to be below 1. But it's important to understand, how this is being measured. This is a calculation. This is a model. This is not a number that we can just go out and measure and say, this is what it actually is. So that's one important thing. And the R0 or R effective varies based on how you model it. And it also varies based on the quality of data that you have. So if you have areas that aren't reporting case numbers or fatality numbers very well, then you got a problem. If you have, areas that aren't testing very much, well, then you have a problem. So it's a quality of data, it's a model, and it's very complicated. And then you have the added problem of it's an average. So R naught over the US is one thing. R naught in any one city or any one district of any one city is going to be completely different. And so some of you might have heard of this term K, and K is sort of the sort of local variation within R effective. And that's also incredibly useful. But all of these are only useful in terms of sort of modeling what's going to happen, what's not happening, how our interventions are affecting things. And that's useful, but it doesn't change the fact that the message is still the same. We're still seeing cases. We know we need testing. We know we need contact tracing. We know we need to wear masks. We know we need to keep physical distancing. So I think in some cases, this focus on exactly what R is has been distracting from a very simple message. And on the other side, it's sort of obscuring what's going on at a local level, because some areas might have R naughts that are R effectives that are well below 1, and then the next town over it might be dramatically above. So it's got to be locally derived, and then it's got to be rapid, because the quicker you can make these calculations, the more effective it is. Some cases, it might be, well, how long does it take the government to release the data? If that takes a long time, then even if you have some interventions, you might be saying, well, okay, people started wearing face masks, but we're not seeing a change in R naught until like a week or two later, because it just takes so long to get the data, calculate the model. So there are all of these limitations. It's still very useful in terms of public health, but I think the point is there are also a lot of other numbers that we can measure. How many cases do we have? How many hospitalizations? What's the intensive care utilization rate? There are a lot of other data that are just as important. So
Okay.
we can get them. Now,
okay.
Where that goes, though, is the interesting part, is saying, okay, I said R naught is an average, and that can be an average over areas, but that can also be an average of events, because I might spread it to 4 people or 3 people, but you might spread it to half a person. Now, what happens when you get a super spreader event? Well, then that's a whole different scenario. So this is where you might have one person that, instead of spreading it to 3 people, spreads it to 100 people, or potentially even more. And so the question is, well, first of all, what effect does that have? And it looks like it has a huge effect. The vast majority of transmission seems to come from a limited number of super spreading events. That doesn't mean that normal transmission isn't as effective, but it just means that those super spreading events amplify that transmission so much faster. It just makes sense. One person infecting 100 is going to spread the virus much faster than one person infecting 4. But why? What is a super spreader? What is a super spreading event? And that is a little bit more complicated, because sometimes that's a person. Sometimes a person can be really effective at spreading the virus. But is that because the person has a really high viral load? Well, it's possible. Is it because that person has a set of behaviors that facilitates a lot of transmission? Not wearing masks, going around, coughing, I don't know. That's possible too. But in a lot of cases, it's a specific event where all of that is combined together. And a really good example is we have super spreading events from, say, there's a great documented one in a choir practice in Washington State. And this is that combination where probably a couple people in that choir had high viral loads. But the practice of singing is all about, projection. And so you are projecting all those respiratory droplets out. You know, if you're singing in a way that isn't projecting those respiratory droplets out, you're probably not singing right, especially not in a choir. So you're projecting all these respiratory droplets out. So this is a behavior and an environment. And then you have a lot of people that are receptive to all that virus coming out, all in a contained environment. So it's that environmental and behavior and possibly high virus load tends to all coalesce together in that sort of right environment or a very wrong environment. And in that case, 87% of the people in that choir got infected from that choir practice. It was incredibly potent transmission event. And now, of course, I worry because in Texas, there was the Pence rally in a church where there was a choir and there was something like 3,000 people there. And, you know, the choir sang and, you know, what did they do? They took the mask down to sing because it was more effective for singing. And then they put the mask back up. But that has potential to be a super spreading event for about 3,000 people. And so it's incredibly possible that a week or two from now, we'll see, you know, maybe 2,500 people in Texas having been infected from that event. And that's how a super spreading event can work. There are a lot of variations, but that's sort of the general idea.
So I think it might be interesting to quickly mention a couple of common super spreader event types. So we've got singing, we've got talking without masks close to each other. That's kind of the social distancing and the masks. So it's like, you know, if you're standing right next to each other, you're 2 feet away, you're kind of projecting your voices at each other. That's going to be far more risky than 6 feet apart with a mask. I can imagine someone coughing on their hand and then grabbing a door handle of like a, you know, mall or a major building where you have to pull the door handle to open the door would be a major cause of spread. That's one of the reasons why you cough into your elbow instead of into your hand, is to reduce the likelihood of that type of event. I think we may be lucky with COVID that it's less surface spread based than it is droplet spread. And so we're lucky as far as we can tell. You know, someone working in a factory that's touching hundreds of objects that go all over, you know, broadly to many locations might be a really terrible form of a super spreader event because it's not localized. Then it can go across the entire world. And on that, we seem to be lucky in that cardboard might be one of the least hospitable environmental surfaces for COVID. So we've got some points of luck with COVID on super spreader events where it's certain types over other types, but that does not mean that other diseases might spread differently. And so are there super spreader events types that stand out to you that would be important with COVID that might give people a good idea of how to understand super spreader events in general so that not only can they modulate their behavior based on the rules of wear a mask, do this, do that, but why doing those things and not doing those things have such a big impact on the spread of the disease?
Yeah, so I mean, first of all, we don't really understand because not every event that, you know, has people singing is going to lead to a super spreading event. So there's a lot more going on in terms of super spreading, whether that's, you know, really high viral loads, something about the immunology of infected people. There are additional elements about what leads to a super spreading event that we don't really understand. And so we can't accurately predict this is going to be a super spreading event or not. But what we can say is that, you know, these are the risks and the environments where it's more likely. And that is, well, if you got a lot of people, then you have a higher risk. If you have a lot of people in a closed environment indoors, that's going to increase the risk. And then if you think about it, you know, the behaviors and the actions that are facilitating transmission, well, you know, we've all had all of the unfortunate meetings of sitting next to somebody and, you know, they are, I don't know, coughing or singing or talking vigorously. And you go and you wipe your face off afterwards because there's just that little bit of spittle or something. And those are all of the things that would be really concerning, because it's not the specific droplets that hit your forehead, it's everything else that's being released into the air. And so it's all of those things. It's sneezing, it's coughing, it's yelling, it's singing, it's, projecting your voice. All of that is putting a lot of these respiratory droplets into the air. And the closer you are to that, the more contained the environment is, the more people producing that. So if you have 10 people that are all shouting compared to just one person, well, the risks are greater. So again, it's all of this sort of additive effect. It's, what's the virus load of the person? How many people are infected? How many people are vigorously talking or yelling or coughing? How close are other people to these people? Are there masks? Are there barriers? Is it a closed environment inside? All of these factors all play together. And it's just like we've talked about risk prevention. Everything that you do is sort of additive and helps. Each one of these risk factors additively predisposes to a significant transmission event.
All right, that's all good to know. It's kind of wear a mask, wash your hands, have some distance between you. Outdoors is safer than indoors. Don't touch stuff that you don't need to touch, and have a mindset of awareness of some of these things, so that you might be able to catch one and maybe stop it before it happens, or just avoid the risk factors in general. A very interesting question that we've had that I've been curious about since I read it is, has there ever been any virus having a beneficial long-term effect?
Yeah, so that's an interesting question, and the short answer is, to my knowledge, no. But I would be shocked. So first of all, there could be, and I just don't know about it. It's entirely possible. There is so much in the field of virology, so I am not the utter repository of virology information here by any stretch of the imagination. But I don't know of any beneficial viruses. Now, this is starting to change, though, because typically the way we've identified viruses has been from disease. Now, other organisms like bacteria or yeast or things like that, they tend to grow on their own. So if you're just saying, okay, what kind of organisms might be living on my skin? Well, then you can sort of swab that and you can look and see what grows and look at it. That's much more difficult for viruses, because you can't just do that and assume that they'll grow. So we've mainly been looking at viruses in terms of causing disease. So it's very possible, we hear about the microbiome a lot. And this is the collection of microbes that sort of live on or in your body. And there's a lot. And we've been learning at how beneficial these microbes are, and absolutely essential they are for so many normal functions of life. The intestinal microbiome, the gut microbiome, this is why all of these probiotics exist. And these tend to be bacterial strains, sometimes yeast strains that live in your gut and help you digest things, help you metabolize things appropriately. They're really important. Now, I would be shocked if there weren't viruses there that also had beneficial effects. The issue is that it's much harder to do those experiments. And we don't have a lot of data on what we call the viriome, the collection of viruses in those environments. We're starting to get that. And we know that there are viruses there. We know that there are viruses that are there that don't seem like they cause any negative effects. But doing those experiments to show that they're beneficial, I bet we will have that data. And I bet that will be true. I bet it's extraordinarily true, but we just don't know that yet because viruses are so much more difficult to work with that field is a lot further behind a lot of other areas of microbiology, whether that's bacteria or yeast or things like that. So I bet there are, we just don't know yet.
All right. Yeah, I had heard a couple, maybe a year ago, someone was talking in kind of the, human maximization, human optimization field. I was having a conversation with her, and she was talking about dieting to modify the gut virome. And I thought that she was misspeaking about gut biome, but she was so confident. I'm like, this can't be. What's really going on here? So I was very curious about the conversation, because like, this sounds completely wild and new and different. And is this like just a misspeaking, or is this actually a thing? And it's like, turns out it's actually a thing. And you can change the viruses spreading through your gut bacteria, because they're actually oftentimes affecting your gut bacteria and not your cells, and being part of that effect. And so changing your diet can affect that, which in turn affects your gut biome. But we don't know much about the beneficiality or interplay of those kind of virome viruses. So yeah, it's definitely interesting, and hopefully we will know a lot more in the near future, because people are definitely looking into it, and there may be some benefits for human well-being.
Oh yeah, huge potential. I mean, and not just gut, but you have a microbiome in your respiratory tract. What effect does that have on diseases like COVID-19? I'm working with some colleagues now that are looking at how COVID-19 changes the digestive microbiome, because COVID can also cause GI infection. And so how does that interplay? And what you eat absolutely changes the dynamics of the microbiome in your gut, and how that interplays with viruses that infect bacteria, viruses that are infecting your cell. These are hugely important questions that we're just starting to scratch at.
All right. What are some common misconceptions that people have about COVID-19? What are some things that you feel like you're saying all the time? What are some things that you hear that you just shake your head and close your eyes and grab your forehead and massage your temples? And what are some wacky and wild things? And all of the above.
Well, I mean, I thought the great, wacky, wild one was that it's actually caused by 5G towers.
5G towers.
That's a fabulous one. That's probably on the highest spectrum of wacky that I've heard. But in terms of misperceptions, there's a lot. I was saying at the beginning that there's this sort of corollary with flu, and that was really useful at first, because first of all, most of our pandemic preparedness of any country was designed against flu. We have huge stockpiles of flu vaccines. We have huge stockpiles of flu drugs, because the perception was that the biggest risk of the next pandemic was a flu pandemic. And I think flu still has the greatest risk of pandemics of most viruses, but it doesn't account for what the WHO calls virus X, which is what SARS-CoV-2 was before, last October. The virus that we don't know about, that you haven't.
It's not the fattest tail. The flu is a known quantity. The flu is a known quantity. It's not the fat end of the tail.
Exactly. And so we were more prepared for flu, and we had more knowledge of how a flu pandemic works, because we just had one in 2009. And so at the start of the pandemic, there were a lot of comparisons made to flu, both in terms of death rate, in terms of transmission, in terms of likely outcome. And I think, and in terms of seasonality, another huge one. And I think because of that comparison at the very beginning, a lot of these misperceptions ended up being propagated. The idea that this is going to be a seasonal disease, the idea that the fatality rate is going to be very low, the idea that the virus might mutate and our vaccines are not going to be applicable. You know, a lot of these misperceptions came from that actually very useful analogy at the beginning, but you know, there's been so much that hasn't been communicated clearly. We've gone from either no information about something or way too much information about something. And so, and of course, it's changed over time. And so that it's been very difficult. So that's been one huge misperception that I think has propagated so many things. You know, we're in July. It's not going away in the summer. It's getting warmer and we're seeing increase in cases. It's because this is not being driven by the seasons. This is a pandemic. Pandemics don't work in the same way. But that was one big misperception, is this sort of seasonality of it. Yeah, so there's just a lot of that stemming from flu. In terms of other sort of misperceptions, you know, I hear a lot of people talking about, oh, it's a mild disease, and if I'm sort of, you know, younger than 70 and don't have any pre-existing health conditions, there's nothing for me to worry about. I'm not at risk. And yeah, you're at a lower risk, but that's absolutely not true. It's just, we've spent so much time focusing on the high risk groups, as well we should, but it doesn't mean that everybody else is a no risk group. It's just a lower risk group. But we see very healthy people in their 20s having critical COVID-19 illness. And so that's another, you know, misperception, is that it's not, you're not safe if you're not in a high risk group. And the other part is that, going along with that, is that, you know, people that get sick, yeah, there's a lot of mild or pre-symptomatic or asymptomatic cases, but COVID-19, even if you're not in that critically ill case where you're in the hospital, this is a very serious illness that lasts a long time and can really wipe people out for a very long time with really protracted recovery, even if you're not needing to go to the hospital. And that's something that's also not talked about a lot, is that sort of, in a lot of cases, that's what we're calling mild. You know, that it's it's much worse than flu disease. So yeah, there's a lot of things, you know, a misperception that we talk about so much and have been combating is that masks don't work. You know, that's a huge misperception that I'm talking about all the time. And it stems from the original discussion on masks, which was saying, don't wear masks. You know, and that was because we wanted to make sure that the medical grade masks were for physicians and healthcare workers that needed them. And because, you know,
I would like to say it wasn't really a we, but some part of the medical virology community, that was the viewpoint. But I think we might be stretching it a little bit. I know you're a part of that community as well, but I think ECV has been very strong from the beginning about wear masks. I know I have. I know that I've sent some rage tweets early on when, you know, some certain highly placed Twitter accounts were sharing very unwise, in my view, messages about not wearing masks. So I think we may be stretching, and I know why you would just say that off the cuff, but I think clarity here may be good, that wear masks. We've always said wear masks.
Exactly. This is a public health, you know,
yeah.
policy debacle.
Yes. And so, yeah, that one, definitely one of the more painful misconceptions. We're coming up on 2 hours, and I think we're not quite at wrap-up time, but we're closing in on it. And I kind of want to do a speed round where people can put in quick questions into the chat or even ask questions on audio if they want. I'll start off with one of the questions we have in the chat. I've heard of some companies using chlorine dioxide tablets in air conditioning units as disinfecting measure. Is this effective?
I mean, you can do a lot of different things to disinfect, airflow, ventilation. It's just, some people, you can disinfect large spaces. This is not something that individual people should do, because you need to seal off the system to make sure that nobody's breathing in chlorine dioxide.
Chlorine, yeah, that's not good.
So yeah, this is not something you do yourself, nor is it ventilation system sterilization, something that seems to be necessary in this pandemic. But yeah, this kind of thing does work, but this is what professional, building disinfectant companies do. And there are lots of ways to do that, and they work very well, but
okay.
not a personal endeavor.
Yeah, one of the things I've heard might be quite effective is using ozone distribution overnight in buildings that aren't in use. Ozone seems to be pretty effective against the virus, especially in conjunction with UV. UV used very effectively, but you have to reach a certain time under light, and so that there's some complications with that.
Exactly. And all good for industrial scale, not relevant for personal scale.
Yeah, it's far difficult to do personally. Let's see. How does COVID-19 compare with the 1918 flu pandemic, speed round version?
Yeah, so the short answer is we don't particularly know, and it's really difficult to compare because we had travel of a world war for the previous pandemic, and we didn't even know what the flu virus was in 1918. So, the data and the quality of the data, it's really difficult to make that comparison. In sheer numbers, we estimate that the 1918 flu was perhaps around as transmissible, but a little bit more deadly. But that could have just been the state of the medical care at that time. It is relatively comparable, but it was significantly more deadly in 1918.
Right. Let's see, what else could we do with speed round? When did you become concerned about COVID-19? December. All right. So you were quite early on this compared with some other people, and generally. So you were watching it from the beginning, then, right?
I didn't know it was going to go like this. Absolutely not. I wasn't concerned at this scale, but I was concerned because there were reports from China of a weird pneumonia and viral infection that seemed to be spreading, coming from a seafood market that seemed concerning, and any new disease that's starting to spread is a real concern. And then watching that start to spread through Wuhan, no, I didn't think it was. I didn't think at that time that it was going to lead to this, but I was worried about it spreading, and maybe being something similar to the previous SARS, but no idea what it was going to turn into. But yeah, I was worried.
All right. Kind of a fun question, or maybe not fun question, but an interesting one at the very least, is what views do you have of the argument that COVID-19 is so successful in humans after its first jump, which raises suspicion, which I think is about the lab leak hypothesis, and kind of if it comes from a lab, there's a number of characteristics of this disease that would make sense in that. It doesn't mean that they're exclusively make sense in that, but that there may be more going on with the origin of this disease than has been publicly acknowledged.
Okay,
let's say.
So maybe we'll do this as two quick
questions.
Sure. One is the origins, and one on the sort of species jump. On the species jump side, we see, flu, swine flu. We call it the 2009 swine flu pandemic was a jump of a virus from a pig to people. It spread incredibly well. It was very effective. So you can get viruses that make that species jump that spread really well. We also don't know what happened in the very early period of time in December, or actually probably in November, or maybe even earlier in China, after the virus jumped into people. It may have been undergoing several mutations that helped it adapt. And, that's why it took a little while before we really started to see a robust infection in Wuhan. But we don't have that old sequence data from the virus, so we don't really know. So you do see some viruses that jump to humans and can transmit really well, but that's not typical. I suspect, and if I was guessing, and this is pure guess, is that the virus was actually circulating in people in, anywhere from August to November in China and slowly adapting. Not a huge number of mutations, because it doesn't necessarily need a lot of adaptation, but that a few mutations that really helped, and then it started to take off. But
right.
I could be
wrong.
And
I guess the second part of that is the lab leak hypothesis side.
Yeah.
We don't have to go in depth, but you are a virologist, and this has been a point of contention. And there's been finger pointing at the virology community. So you have a point to defend yourself, or maybe to take a stance of more skepticism than has been popular.
So unfortunately, this is one of those things that unless we actually get confirmation from the lab that there was a release, you're never going to be able to definitively prove or disprove it. All the best genetic evidence that we do on the virus suggests that, in fact, it evolved and emerged naturally. We see close relatives in bats and pangolins. There's a very convincing genetic argument that it's not a lab strain and that it wasn't an accidental or intentional release. But you can't definitively prove that, because those same evolution that we're inferring to occur in the wild could have, of course, occurred in a lab. We know that there were labs in Wuhan that were working on bat coronaviruses that are related. So is it possible that it was an accidental release? It's possible. There's no evidence that says it was, whereas there's evidence that implies it was naturally occurring. But this is something that I hate talking about, because it's a great question, so I'm happy to talk about it in this context, but I hate talking about it in general because it's just placing blame. There's no, yes, in the long term, evaluating, lab biosafety, great. Let's have that conversation. But right now, we need masks, we need public health, we need testing, we need that infrastructure here, now, weeks ago. So talking about
months ago
is just this is distracting from what we need to be talking about right now. Maybe later it will be useful, maybe later we can get into it, but right now it's just distracting.
So I guess I'd like to follow up slightly, because I think it may be important to follow up in this. Is one of the points made by some of the people who are the most serious about the lab leak hypothesis, like, as I believe it's Yuri Dagan, who wrote the sort of the major Medium article that kind of went the most in depth about exploring the potential of a lab leak and what evidence there's for it, which I haven't read his Medium article, but he claims he started by looking for arguments against it and was converted by his inspection of the evidence, which makes a claim that there's evidence there, but I haven't read it, so I can't analyze it. But that if we know or suspect that it is a lab virus, that might change the dynamics of how the virus behaves, because the selective environment of a lab is going to be different than the general population or a transfer from a wild species. And that knowledge, if we had it and we had the certainty of it, could allow us to affect the disease more effectively because we'd be able to leverage our knowledge about it that, we might not have just from general observation. And I think that's the argument for arguing about this.
And I completely disagree with that argument. I think that it's a great, it's a great question, and it's a great setup to having this argument. But in fact, the virus is so far from when it emerged in people, whether that came from a lab or not. It's already had a little bit of adaptation already. Some of you might have seen this paper about the single point mutation in the spike protein that was just published that we've been hearing about for a long time. There have been adaptive mutations already. And so knowing if it was engineered in the lab tells us absolutely nothing more than if it came from the wild right now, because we have the genetic sequence. We see how it compares to SARS and MERS. We see how it's behaving in people. Where it came from really doesn't tell us anything useful right now for public health right now. It doesn't change anything, because short of them having that virus and a matched vaccine in the lab, which I don't think is the argument, but obviously if they had a vaccine or a special drug that worked for it and they weren't releasing it, then it would be a different story. But there's absolutely no evidence for it. And the evidence for the lab release is just coming from the fact that there were labs there that were working on similar viruses and in bats, and so that there's the possibility. But my statement against that is lab science is 100% driven by publications. And this is just as true in China as it is in the US. And so if that lab had found SARS-CoV-2 in the wild and then were working with it or adapting or anything like that, that would have been a huge publication. And that would have done really well for the lab, for the Institute of Virology in Wuhan, and for China as a whole. There would have been a lot of incentive to publish this, and it never was. Now, you can get into all sorts of conspiracy theories, and is it possible? Sure, it's possible. It's going to be very difficult to prove or disprove, but there's not solid evidence to say that it came from the lab.
All right. I'll take that answer. That's a good answer. I don't think that we need to pick into that any deeper, but we can shift over to another politically charged question. Sort of backloading this a little bit here is we have a question asking, how or why did the HUD and CDC drop the ball with COVID? Because it seems that we've had a very rocky relationship with sort of the major public health entities who, I think, inarguably have said some things that probably should not have been said and have definitely not been leading lights of clear communication without backtracking and such. So I think that's a valid question and frame for it is why and how have things seemed to have gone so wrong with the public health, the professional public health response, not just the political response.
So you were saving the easy questions for last, I see.
Yeah.
Right.
And I guess I'll say it so that we can have confidence as a community, and me as your host is we don't have to publish put on YouTube this last segment if we decide not to. I think that's an open decision, and part of the reason why I've saved these more medical, more divisive questions for the end is so that it's an easy cut if we want to cut it. But I wanted to ask these things because there's things that I know some of us are very interested in, and I'm certainly interested in. I think they're good to have as part of this discussion, even if it's just us on this live stream.
No, divisive questions are good. These are questions that are relevant, people want to know and are important. So I think there are two sort of separate answers here, because I don't think you can lump the WHO response into the same bin as the CDC response. So
Okay.
on the WHO response side, you know, from the US, we've heard so much WHO bashing about how bad they've done, and you know, how they've responded to China and things like that. Now, from my perspective, as a virologist looking at public health, I thought the WHO has done actually a very good job. Could they have done better? Sure. Retrospective analysis is perfect for that. Have there been some missteps? Sure. There's no way you're not going to have any missteps in a pandemic response. But one of the biggest criticisms against the WHO is, you know, the information flow from China. And to an extent, this is not really a fair criticism of the WHO, because the WHO response was limited based on the data that China was providing. It's not the WHO's response to collect the data and to go to China and get that data. China has the public health response. They're managing their outbreak and reporting to the WHO. So, you know, this criticism, can you criticize China's early response and their information flow? Absolutely. Does that then translate to the WHO's early response? I don't think so. Did it limit their response? Yeah, but I don't think that it was because of WHO policy that was what affected it. So, and you know, you also have this issue of, you know, people saying, oh, they should have taken a stronger response with China. But the WHO has to manage public health relations with countries around the world. They can't afford to go and burn bridges. They need to have that working relationship with everybody. And so could they have maybe been a little bit stronger? Sure. But is it the fault of WHO? I don't think so. If you look at WHO messaging, they have been very proactive. They have been very clear. And yeah, their message has changed, but their message changed when the outbreak changed. You know, when it reached the new threshold, and they're saying, well, you should have declared it a pandemic earlier. Well, they were waiting to see that it was in fact a pandemic, because the WHO was criticized in 2009 for declaring swine flu a pandemic too early. So, you know, it's a balancing act. And I think overall they did very well, and they have been providing such strong messaging from the very beginning, saying, look, you need to test, you need to trace, you need to isolate, and you need to have public health responses in place. And you need to do all of it. Not just one is enough. And with that, you can contain it. This is what ECV has been saying the whole time. We can contain, we can stop the spread, we can get to zero. So I think the WHO has actually done a very strong job. Now, the CDC, I think this is all
before we go to the CDC, I think I'd like to dig into the WHO thing just a little bit. I appreciate that you've given a strong defense of WHO, given the context that they must operate under. And I think that a point that I would make in contrast is perhaps the model that we have in general that the WHO operates underneath, so the meta model, as a civilization versus as like an organization or as sort of a current global system, may be far from optimal. And that given the situation that they operate under, they did well, which is a different assessment of the situation that they operate under should be changed because it doesn't actually solve the problem in a very highly globalized world where a disease outbreak can spread to nearly every country in the world within weeks. And that the response models that we currently have, the expectations that we have, the way that the global economy and governmental leadership interfaces with pandemic response as a field, and the science and the preparedness that we have are all not up to the current task. And that I think is a different assessment to, did the WHO as an organization handle this immediate situation given the current overstructure that they must operate within. I think those are two different analyses, and I would be far stronger on my second analysis, that the underlying models that we've built these things on may have worked in the 60s and the 70s and even the 80s, but in the modern hyper-connected digital era with so much global travel, these things no longer are up to the task. And so what are your thoughts on that analysis, that we need to rethink the underlying model, given that you know far more about the field than I do?
Oh, absolutely. I completely agree with you on that. That you know, from that perspective, yeah, the response was not good, but you know, given the constraints, that's what I was referring to. But I think you're absolutely right. The model that, you know, we're operating under is not a global integrated model. It's not a model that accounts for climate change. It's not a model that accounts for, it's not what we call a One Health model, where we're looking at how all of these parameters come in to affect global health. We need to think about global health. We see in this pandemic, we can't just think about one country. We can't just think about one aspect of health at a time. This is all interconnected. And do we need a different model to really account for this? Absolutely. Is that an easy thing to do? Absolutely not. But maybe this will be a little bit of the motivation to actually start to rethink how we respond to infectious diseases, how we respond to climate change, how we respond to risks. You know, this is not going to be the only pandemic. You know,
Right.
you know, people are worried about the next flu pandemic still. There are risks of new viruses emerging all the time, or bacteria. You know, this doesn't have to be just viruses. And we need to be able to respond to that globally. And we're not responding to that globally. We're a collection of individual responses responding to a collection of individual things. And that's not as effective as it could be, but that's a huge model and system to change.
Yeah, and so that's kind of some of the risk management or risk awareness field that's partly recognizing the importance of sustainability of the models that we build, not just environmental sustainability or economic sustainability, but sort of we need to build stuff that works reliably, iteratively across events, which is kind of a point to what you said about the WHO being hit for swine flu, and so being more cautious this time. That's not a sustainable model. You can't ping around based on the whims of the public. You need to be able to hold a firm line and such and so on. It's a very complicated thing, but I think that we can do better, but we may have done the best that we could with the old breaking down, no longer oriented towards the world that we now have models. So I think we did well there. And then the CDC, if you want to go into that some.
I mean, on the CDC side, this is, I mean, it's not just the CDC. I'd say, you know, the U.S. public health response overall has been a failure to lead. It's been pretty clear. I'm not going to touch on, you know, the political sides that necessarily underlie that, outside of saying there has been a resistance to acknowledge what was happening. There's been a resistance to taking charge and investing and committing to the public health infrastructure that's necessary to combat this. And there's been a resistance to proactive, protective messaging to the community. And then there's been swings in messaging that's been fairly consistent swings in messaging that's occurred through the duration. So I think it's a, it's a failure to lead, and not necessarily of any, not saying that that's a specific failure of the CDC to lead, but a failure of the US public health response to lead and to do public health response. I mean, the US goes to countries around the world to assist with other outbreaks and epidemics. The US CDC went to the DRC and to West Africa to combat the Ebola virus epidemic, went there and used the public health expertise that we have and that we know. But we didn't do the same thing. We weren't willing, and again, I'm using this sort of larger, we weren't willing to commit and realize that that risk is applicable to us. I think there was a sense of invulnerability, that this is something that happens to other countries. This is not something that happens to the US. And that we have such the public health infrastructure, such strong research, such strong expertise that we would be resistant, that this wouldn't happen in the US. And so regardless of the political will that led to where things went, this is a failure to lead and a failure to act and a failure to acknowledge what was happening and what still is happening.
All right. Thank you, Jeremy, so much. I think it's time that we wrap up. If anyone has like a final question that they would like to ask, or if Jeremy, you would like to say some final words, and certainly you can plug yourself before we go out. But I think this is a good spot to end this. So, Jeremy, thank you again. Really appreciated it. This was an excellent conversation and talk. We had some great questions from the team here following us. Most of the question that I asked was from them. I'm not quite as creative as our group chat, and so I want to thank them for asking excellent questions and for staying with us for, what is it, almost 2.5 hours of talking about virology. So thank you, Jeremy.
Can I ask a question?
Yeah.
Go ahead,
Yeah.
Shantaka.
Yeah, I met Jeremy Rossman. We had a meeting along with Gary a few weeks ago at the beginning, I believe. Sorry, I had to take care of some family things. So I joined late.
No problem.
Type A, is that the most vulnerable, the blood type A for COVID?
And O is the most resistant. Well, I don't know if resistant is quite the right word, but O is the least, yeah.
Oh.
Yeah.
Okay, that's my question.
Yeah, the A and O seems to be the two sides.
So A is bad then?
Yeah, we don't know how, we don't know exactly why, but yeah, A seems to be more prone and O seems to be more resistant.
So to make it more resistant, is there anything we can take to change the blood?
No.
Not the type, but I mean resistance, no.
No, because we don't really understand. It could just be correlated with some other factor. We don't really understand why that blood type is affecting severity of disease. So unfortunately, no, there's nothing that we can't utilize that yet in treatment. I mean, maybe somebody will figure out something in tomorrow or a year that will allow that, but right now, unfortunately, no.
Okay, thank you. Appreciate it. Very good. Excellent.
Thank you. And anybody else, if anybody has any other questions, I'm happy to take them.
Sure.
Yeah.
And sort of the tag-on question to that, is there a potential prophylactic that might be within reach, that might be trade-offs wise worth using for a couple months on the potential that it might be protective, but we don't know yet and or haven't been testing to see if it is. Are there candidates that come to mind, or is it sort of is it a dead field at this point?
Yeah, no, not the answer is no. Might we get one? I mean, there are a lot of drugs that are being trialed, so it is possible that one will work as a prophylactic. It's entirely possible. There are a lot of drugs being tried right now, a lot of potentials. But no, nothing looks promising for being a prophylactic at this point in time. Absolutely is nothing is worth taking right now. Also, it's been very difficult to get, prophylactic drugs that work against viruses. We don't have many of these. You do have the PrEP for HIV. That's a combination of drugs, and that does seem to work. But look how long HIV has been around, and this is a pretty new development. So it's taken a long time to have a preventative, and it's not something that you want to take all the time. So, I'm not optimistic that we're going to find one. We still don't have a drug that prevents you getting flu. So it's possible. I hope so, but yeah, we don't have one yet, and nothing that I see on the horizon. I think it's much more likely that we have a vaccine before we have a preventative therapeutic.
Okay. Does quinine affect any viruses? Just out of curiosity.
Yeah, I mean, I've never heard of quinine specifically being tested against viruses because,
or something from that family. Yeah, there's something in that family. But if it hasn't sort of been known to be effective before, but it's sort of one of the things that is known to be a decent prophylactic for a number of things, and that's why we're playing with it, because we're out of ideas in some sense. I guess that was one question. Another question that we've got now is vitamin D. What are your thoughts on the vitamin D data that we have about this disease?
That's one that should be, I don't know if it's exactly a misperception, but that's one that is something I almost wish this had come up earlier, because I think this is a really good question that I've heard a lot about, because vitamin D is one of those things it's just so easy. It's so easy to do. And so there's a lot of talk about saying, oh, this is the miracle cure, and all we need to do is treat with vitamin D and we're fine. And that's the misperception, because vitamin D is not going to cure the virus. This is not a therapeutic, but making sure that you're not vitamin D deficient can make a fair difference. So there are a lot of things that you can do personally that help you mitigate the either chances of being infected or how your body responds to being infected. So, being physically fit, getting exercise, making sure that you're, not vitamin D deficient. Now, you don't want to give too much, because you can overdose on vitamin D. So that's not good. You got to be careful. You can have, and more vitamin D isn't better than the right amount of vitamin D. But vitamin D deficiency is associated with an impaired immune response. And so you can have increased risk with lower vitamin D. Now, it's not a perfect correlation, and it's not like if you have your vitamin D, you're not going to get infected. Absolutely not. But it's like if you're being physically fit and you're getting vitamin D and you're eating healthy, all of this again, this is the additive thing. All of this helps. And, it's not like it's a yes or no. It's not like if you have a vitamin D supplement, you're not going to get infected or have severe disease. You could absolutely. And it's not like if you don't have vitamin D, you're definitely going to infect it. You might not. But it's just one of those things that does help some.
All right. Is there a protocol that you would suggest on that sort of, if, reduce your risk of severe disease, kind of as a prophylactic approach to better health during the COVID end times?
Yeah, it's really straightforward, and it's exactly what everybody's told us all for a long time is, get exercise. The more exercise you get, the better. If you can exercise for a half an hour a day, that's great. Get some exercise and eat well. Balanced diet, not a lot of fatty foods, not a lot of salt, not a lot of sugar, balanced vegetables so that you get all your vitamins. Spend some time outside so you get some more vitamin D. That's what we're talking about. It's nothing complicated. It's everything that we've been told for a long time.
Vitamin C play into that much?
Vitamin C also, absolutely. But again, what we're talking about more is deficiency versus having enough. We're not talking about overload, because overload of any vitamin can be bad. So we're just talking about making sure that you're not deficient, because if you don't have enough vitamin C, if you don't have enough vitamin D, that can cause problems. But making sure that you're eating the balanced diet so that you do get enough of the basic vitamins, making sure that you're getting that, 100% of the vitamins a day. You don't need 1,000%. You don't need 200%, just the 100. It's just basic. That helps. It's all contributing to how effectively your body is responding. Get sleep, that's another thing. Reductions of sleep can impair your response. Extra stress can too. So, none of us are really in that lots of sleep, no stress category. So we got those risk factors here. But, again, it's that exercise, diet, not having the gin, I guess, unfortunately, not having the stress, sleeping well, all of that helps. And again, there's no one thing that makes or breaks it. Yeah, it's been really fun chatting. I'm happy to do this again. If people have more virology questions or want to, go off on deeper tangents, we can do that in the future. But yeah, thanks Gio for putting this on. This has been fun. I mean, my research and humanitarian aid organization website is researchaidnetworks.org. We have a COVID page on there and post a lot of things on Twitter, which is just @researchaidnet. So yeah, happy to have you follow me.